I've been practicing this word for a while. Parthenogenesis. Parthenogenesis. Parthenogenesis? One sec.
Parthenogenesis.
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 Nice.
Anyways, we've seen it in movies, shows, and other media. But what is it exactly? Essentially, parthenogenesis. Try saying that 5 times. Okay. PG is a type of asexual reproduction when an unfertilized egg grows into a full organism. Creatures that do this skip the whole dating thing, and the babies produced are genetic clones of their mother. PG happens in bug kingdoms like ants and bees, and reptiles and fish kingdoms, but not as often as those two. Also, how bugs do PG is a little different than how other animals do it. They lack a sex chromosome. Weird, right? Some species really said nope to the whole X and Y thing. Now, before we get too deep, let's talk about the reproductive process for sexually reproducing organisms. Meiosis. It's the cell division process that's responsible for making gametes, aka sperm and egg cells. To explain the process is a whole video in itself. But I don't have time for that. So here's the rundown for meiosis in humans. Humans have 46 chromosomes, or 23 pairs. These are called diploid cells, because they have 2 sets of chromosomes one from each parent. Gametes are called haploid cells, because they have 23 chromosomes, or 1 set. A diploid cell goes through interphase, where it duplicates its DNA. Once that's done, the cell goes through 4 phases, called prophase, metaphase, anaphase, and telophase, or PMAT for short, because I don't want to repeat all that. PMAT doesn't happen once. Oh no. The cell goes through PMAT twice, or better known as meiosis 1 and meiosis 2. After going through meiosis 1 and 2, the cell has divided to the point where it is now left with 23 chromosomes. Half the usual amount, but it's mixed in a unique way. Without this process, there would be no biodiversity on Earth. No life, no awkward family reunions, or no Shrek 5 trailer. You know what? Actually, that wouldn't be too bad. I mean, look at this. Oh my, ew. Anyways, meiosis is a big deal, and it's why we aren't exact copies of our parents. It allows for so much genetic variety. But PG skips meiosis entirely. So, could humans do it naturally? Kind of. But instead of a baby, it's kind of a little scary. You have been warned. Do not Google images of this. If you do, you might be scarred like I am. Because it is absolutely disturbing. When PG occurs spontaneously in females, it results in what is called an ovarian teratoma. There are a few types of teratomas, but for this video, let's just focus on this one. Now, remember, in meiosis, diploid cells are turned into haploid cells. A theory suggests, because scientists actually don't know why teratomas occur, but a theory suggests at the end of the second phase of meiosis, when the cell gets rid of the other 23 chromosomes, becoming a haploid cell, those 23 chromosomes reemerge with the egg, restoring itself back to its diploid self, I guess. This adds up to the correct 46 chromosomes needed to make a baby, right? It instead makes up some biological horror story, kind of looking like you. Just kidding. Unless. So yes, while the numbers check out, genes are tricky. Without the use of paternal genes, there's no proper genetic regulation. Things don't go smoothly. Instead, you get a tumor with various types of cells that can grow hair, or muscle, or even teeth. Ugh. Telling you, don't look at it. Essentially, it's basically the universe saying, yeah, humans aren't meant to do that. At least, not naturally. In the past, induced PG in mice and monkeys resulted in abnormal developments. Prior research to force mammals to reproduce via PG have failed because of genomic imprinting. Mammals have this sort of imprinted genetic region where either the maternal or paternal chromosome is inactivated in the offspring. Think of like a on and off switch. That's essential for the development process to proceed normally. A mammal developing from PG would have a double dose of maternal imprinted genes and like the imprinted paternal genes, right? This then leads to the abnormalities. Hence why, naturally, an ovarian teratoma is the result. So, what happens if science gets involved? Because messing with nature never had any type of ethical or moral dilemma like ever in the history of humans, right? A study published in March of 2022 showcased viable offspring derived from a single unfertilized mammalian octote in mice. There's a lot to go over in this, but to save some time, I'll give you the SparkNote version. Do people still use that? In mammals, PG is limited because of the problem arising from genomic imprinting, like I mentioned earlier. A viable offspring was achieved by the targeted DNA methylation, rewriting of 7 imprinting control regions, 2 of which were able to trick the egg into thinking it was a sperm. Weird. There were 387 modified embryos, and only 3 mice pups were born. 2 out of 3 were born but showed very low body weight and stunted growth, and sadly died within 24 hours. The last one made it to adulthood, but it was, forever fun-sized. It too showed growth issues. It was almost 20% smaller than mice of its age, due to the scientists not fixing a gene involved in postnatal growth. And when this PG mouse had mice pups with a male mouse, her babies were totally fine. Essentially, nature kinda decided to hit the reset button on her genes before passing them on. And so scientists tried this over again. But this time they fixed the gene for postnatal growth, and this resulted in 2 viable mice with no growth issues. Now, you could make the argument that this wasn't true Parthenogenesis, but a viable mammal was produced without the use of sperm. So yay, success in mammals giving birth to PG offspring. But what does this mean for humans? Well, we're not going to be cloning ourselves anytime soon. But apart from that, I really have no idea. I'm not a scientist, nor am I some ethical advisor for the world. But what I do know is that it's a very fascinating subject that needs more study. And it poses a lot of ethical and scientific questions, especially if it's used on humans. I mean, if true PG were to happen with humans, what would happen to every aspect of humanity? I mean, our genetic diversity would definitely lag. We'd all be clones, and, well, that's boring. But everything else that makes us, well, us, what would happen? Well, I guess time will tell.